He had eaten a bacon and egg sandwich every morning for 40 years, grilled chicken breast almost every evening, a diet that by every conventional metric was reasonable, high protein, moderate fat, not excessive in calories, far better than most of what his peers were eating. He had never been significantly overweight. He had no diabetes. His cholesterol was in range. When he came to his appointment at 71, complaining of fatigue, joint stiffness, cognitive slowing, and a general sense that his body was deteriorating faster than his age should predict, his blood work was reviewed, and his physician told him there was nothing specifically wrong. He was in the normal range on everything they measured. What his physician did not measure, what almost no routine blood panel measures, was the level of advanced glycation end products in his tissue. It would have been elevated. It would have been elevated by decades of grilled bacon, brown chicken skin, toasted bread, roasted vegetables, and every other food that had undergone the chemical transformation that happens when proteins and fats are heated above 150 degrees Celsius in the presence of sugars. The compounds produced by that transformation were accumulating in his tissues, binding to specific receptors on his cells, activating the inflammatory cascade that was driving his mitochondria toward dysfunction, accelerating the shortening of his telomeres, and prompting his cells to enter the senescent state, the zombie state in which damaged cells refuse to die but release inflammatory signals that damage their neighbors, at a rate that research now shows is measurable, predictable, and substantially reducible by a single change in how food is prepared. The compound I am going to describe today is not a food ingredient you can look up on a label. It is not something a food manufacturer adds to a product. It is something that forms in the food you are eating as a consequence of how it is cooked. It is present in high concentrations in grilled meats, fried foods, roasted nuts, baked and toasted carbohydrates, pasteurized dairy products, and the browning on virtually every processed food that has been heated during manufacture. It is called a dietary advanced glycation end product, a DAGE, and the research on its relationship to cellular aging, mitochondrial function, chronic inflammation, kidney disease, cardiovascular aging, and cognitive decline is among the most consequential and least communicated findings in nutritional gerontology of the last 2 decades. I am Dr. Edward. What you are about to hear is educational information based on published scientific research. It is not a substitute for personalized medical care. The dietary modification I am going to describe is evidence-supported, low-risk, and accessible without any special equipment or expensive ingredients. But the biology I will explain is complex enough that I want you to understand the mechanism fully before I give you the intervention, because understanding why the change works is what makes it stick. Subscribe to this channel right now before we go into the chemistry. What I am about to describe changes not just what you eat, but how you prepare it. And the research behind it will permanently change how you interpret the smell of browning food. Subscribe now. It is free. And stay with me. Let me start with the chemistry of how advanced glycation end products form, because the name sounds technical and the mechanism is actually straightforward once you understand the basic reaction. Glycation, as distinct from glycosylation, which is enzymatically controlled, is the non-enzymatic reaction between a sugar molecule and a protein or fat. It happens spontaneously whenever a reducing sugar comes into chemical proximity with a free amino group on a protein or with a polyunsaturated fatty acid in a lipid. The initial product of this reaction is a relatively unstable compound called a Schiff base, which rearranges into a more stable form called an Amadori product. Over time, through a series of further rearrangements and oxidative reactions, Amadori products undergo additional transformations that produce the final, stable, largely irreversible compounds called advanced glycation end products. The most studied and most clinically relevant dietary ages include carboxymethylicin, CML, formed from the reaction of lysine residues in proteins with oxidized lipids and sugars, methyl glyoxyl adducts, formed from reactive carbonyl compounds produced during sugar and fat degradation, and pentosidenine, a fluorescent age formed in long-lived proteins like collagen. This same glycation chemistry occurs in 2 contexts. It occurs endogenously, inside the body, when blood glucose is chronically elevated and glucose reacts with body proteins over time, producing the hemoglobin A1C that is measured in diabetes management, the glycated LDL that contributes to atherosclerosis, and the retinal glycation that underlies diabetic eye disease. And it occurs exogenously, in food, when proteins and fats are heated in the presence of sugars, accelerating the Maillard reaction that produces the browning, the crust, the caramelization, and the deeply appealing flavors and aromas that humans find so compelling in cooked food. The Maillard reaction, named after the French chemist Louis-Camille Maillard, who described it in 1912, is responsible for the color and flavor of roasted coffee, grilled meat, toasted bread, baked cookies, and virtually every cooked food that humans find palatable. It is also responsible for generating the dietary ages that are absorbed through the gut, enter the circulation, and activate the cellular receptors that drive the accelerated aging biology I am going to describe. The critical insight is that the ages in food are not simply analogous to the endogenous ages formed inside the body. They are substantially the same chemical compounds, and when absorbed from the diet, they add to the body's existing age burden rather than being treated as foreign molecules and neutralized. Research has found that approximately 10% of dietary ages are absorbed across the intestinal wall, enter the circulation as free age adducts, and are distributed to tissues throughout the body. Some are cleared by the kidneys. A significant proportion are deposited in long-lived tissue proteins, the collagen of skin and blood vessels, the crystalline proteins of the eye lens, the myelin proteins of nerve fibers, where they accumulate because these structural proteins turn over very slowly, and the ages attached to them persist for years or decades rather than the hours or days that more rapidly recycled proteins remain in circulation. The mechanism by which dietary ages damage cells operates through a specific receptor called RAGE, the receptor for advanced glycation end products. RAGE is expressed on the surface of many cell types, including vascular endothelial cells, macrophages, neurons, cardiomyocytes, and tubular cells of the kidney. When dietary or endogenous ages bind to RAGE, the receptor activates intracellular signaling cascades, specifically the NF-kappa B pathway, the primary master regulator of inflammatory gene expression in mammalian cells. NF-kappa B activation produces a cascade of pro-inflammatory cytokines including interleukin-6, tumor necrosis factor alpha, and interleukin-1 beta. These cytokines are the same mediators that drive the chronic low-grade inflammation that researchers call inflammaging, the age-related inflammatory state that underlies cardiovascular disease, cognitive decline, sarcopenia, insulin resistance, and virtually every age-related chronic disease. RAGE activation is, in this sense, a direct molecular bridge between dietary age consumption and the inflammatory biology of accelerated aging. But the RAGE NF-kappa B pathway is not the only mechanism by which dietary ages damage cells. Research from the laboratory of Dr. Helen Vlasara at Mount Sinai Hospital, the scientist who has contributed more to the understanding of dietary ages than any other researcher in the field, has established that dietary age consumption directly impairs mitochondrial function through a mechanism involving the oxidative modification of mitochondrial proteins and the impairment of electron transport chain complexes. When mitochondrial proteins are glycated, either by endogenous ages or by dietary ages that have been deposited in mitochondrial tissue, the efficiency of oxidative phosphorylation falls, the production of reactive oxygen species at the electron transport chain increases, and the mitochondria enter a dysfunctional state characterized by reduced ATP production, increased oxidative damage to mitochondrial DNA, and eventually mitochondrial membrane permeabilization that triggers apoptosis. Programmed cell death. The research quantifying this effect is the source of the claim in the title of this video. Studies published in the journal PNAS by Vlasara and colleagues found that mice fed a diet high in dietary ages showed mitochondrial dysfunction and cellular senescence rates approximately 2 to 3 times higher than mice fed the same caloric intake with the same macronutrient composition, but prepared in ways that minimized age formation. The age-restricted mice showed measurably longer median lifespan, lower inflammatory marker burden, better kidney function, improved insulin sensitivity, and reduced rates of the neural degeneration that in higher-age animals produced Alzheimer's-like pathological changes. The 2 to 3 times figure refers specifically to the rate of mitochondrial dysfunction and cellular senescence accumulation, both of which are direct drivers of the biological aging rate I have described in previous videos on autophagy and the longevity switch. Subscribe to this channel right now because I am about to give you the specific foods and cooking methods ranked by age content. Information that turns an abstract chemistry lecture into a specific grocery list and a specific set of cooking modifications. Subscribe, it is free, and let us continue. Research from Dr. Vlasara's laboratory published in the Journal of the American Dietetic Association and replicated by independent groups in the United States and Europe has measured the dietary age content of thousands of common foods using standardized assay methods expressed in kilunits of CML equivalents per gram of food. The findings are counterintuitive in important ways and deserve careful attention because they challenge some of the most deeply held assumptions about what constitutes healthy cooking. The highest age content foods are not the foods traditionally considered unhealthy for other reasons. Processed meats, bacon, sausage, hot dogs are among the highest, with bacon generating approximately 91,577 kilunits of ages per 100 gram serving when pan-fried or grilled. Grilled chicken skin reaches similar levels. But what surprises many people is that several foods considered healthy produce very high age concentrations depending on preparation method. Grilled salmon produces substantially more ages than salmon poached or steamed. Roasted almonds contain approximately 6 times the ages of raw almonds. Toasted bread contains 3 to 4 times the ages of untoasted bread. Roasted coffee, which is the form consumed by virtually every coffee drinker, has undergone the Maillard reaction extensively during the roasting process, and while the age content in the brewed beverage is diluted, the cumulative daily exposure from multiple cups adds meaningfully to total dietary age load. The pattern in these numbers is consistent and mechanistically logical. The higher the cooking temperature, the lower the water content of the cooking environment, and the longer the cooking duration, the more ages are generated. Dry heat, grilling, broiling, roasting, frying, baking, produces dramatically more ages than moist heat. Boiling, steaming, poaching, slow cooking with liquid at equivalent temperatures. This is because water acts as a competitive sink for the reactive carbonyl compounds that drive age formation. When food is cooked in water, those reactive compounds preferentially react with water rather than with proteins and lipids. When food is cooked without water, in a dry oven, on a hot grill, in hot oil, the reactive compounds react with the food's own proteins and fats and form ages at rates that can be 10 to 100 times higher than the same food prepared with moisture. The practical implications of this chemistry are specific and immediately actionable. A chicken breast boiled or poached in broth with aromatics contains approximately 1,000 to 2,000 kilunits of ages per serving. The same chicken breast grilled until it develops characteristic char marks contains approximately 8,000 to 10,000 kilounits. The same breast fried in oil for equivalent duration contains approximately 9,000 to 11,000 kilounits. The protein content, caloric density, and macronutrient composition of all 3 preparations are essentially identical. The age content differs by nearly a factor of 10. From a cellular aging perspective, these are not nutritionally equivalent preparations of the same food. I want to address the kidney clearance piece specifically because it is particularly relevant to the senior population, and it explains why dietary age exposure is not a self-correcting problem that the body simply manages. The kidneys are the primary route of age clearance. Circulating age compounds are filtered at the glomerulus and excreted in the urine, and this clearance mechanism keeps circulating age levels within a manageable range in young adults with healthy kidney function. But as kidney function declines with age, and as I described in the foamy urine video, GFR falls progressively through the 60s and 70s in most adults, age clearance efficiency falls proportionally. A senior with a GFR of 60, which would not qualify as kidney disease on most clinical scales, is clearing dietary ages at approximately 40% less efficiency than a 25-year-old with a GFR of 100. A senior with a GFR of 45 is clearing them at less than half the rate. The same dietary age intake that a younger adult metabolizes and excretes efficiently is accumulating in the tissues of a senior whose kidney clearance is age diminished at concentrations that the body's rage-mediated detection and neutralization systems were not designed to handle continuously. This is the compounding vulnerability that makes dietary age restriction so specifically urgent in the over 60 population. The dietary age exposure from a lifetime of grilling, frying, and roasting is arriving at tissues that have progressively less capacity to clear it, with rage receptors that are progressively more sensitized by decades of accumulated age binding, and with mitochondria that are already under higher oxidative stress from the intrinsic metabolic changes of aging. The 3 times faster cellular aging described in the research does not occur immediately or uniformly. It accumulates over decades of daily age exposure and reaches its most significant biological expression precisely in the age range, 60 to 80, when the cellular consequences of that accumulation become visible as the accelerated aging we attribute to getting older. The human clinical trial evidence for dietary age restriction is not as extensive as the animal model data, but the existing human research is compelling. A randomized crossover trial published in the journal Diabetes Care assigned adults over 60 to either a standard American diet or an isocaloric diet with significantly reduced age content achieved through cooking method modification rather than any change in food composition. After 4 months, participants on the low-age diet showed significantly reduced serum CML levels, reduced TNF-alpha and IL-6 inflammatory markers, improved insulin sensitivity measured by HOMA-IR, and measurably improved kidney function as measured by cystatin C-based GFR compared to their baseline on the high-age diet. The same foods, the same macronutrients, the same caloric intake, different cooking methods, measurably different cellular, inflammatory, and metabolic outcomes. A separate clinical study published in the Proceedings of the National Academy of Sciences examined the effect of dietary age restriction on oxidative stress markers and telomere length in older adults over 12 months. Adults randomized to the low-age preparation method showed significantly reduced plasma 8-isoprostane, a marker of lipid oxidation, and significantly better preserved leukocyte telomere length compared to those on the standard high-age diet. Telomere length is the molecular clock of cellular aging, the protective cap on chromosomes that shortens with each cell division and with oxidative stress, and whose rate of shortening directly determines how quickly cells approach the senescence threshold. The dietary age intervention was not reversing telomere shortening, but it was measurably slowing it by reducing the oxidative stress that drives telomere attrition independent of cell division. Now I want to address the interaction between dietary ages and the ultra-processed food consumption that characterizes the modern senior diet, because the commercial food processing industry has created an age exposure pathway that most people never consider because the cooking appears to happen before the food arrives at the table. Ultra-processed foods, defined by the NOVA classification system as industrial formulations containing ingredients not typically found in home kitchens, including preservatives, emulsifiers, artificial flavors, colors, and processing aids, are almost universally high in dietary ages because they are manufactured using the high-heat processing methods that maximize palatability and shelf life. Commercial baked goods undergo the Maillard reaction during baking. Extruded breakfast cereals undergo it during extrusion. Roasted and salted snack foods undergo it during manufacture. Cured and smoked meats undergo it during processing. The age burden in a typical senior's diet is not only from the grilling and frying they do at home, it is compounded by the age content of every packaged, processed, and manufactured food product they consume, most of which have been subjected to high-heat processing multiple times during their manufacturing journey. Research published in the British Journal of Nutrition found that adults consuming the highest proportion of ultra-processed foods had significantly higher circulating age markers than those consuming the lowest proportion, independent of total caloric intake, macronutrient composition, and body weight. The ultra-processed food age burden was attributable not to any specific ingredient in the products, but to the processing method itself, a finding that explains why two people eating the same number of calories from equivalent macronutrients can have dramatically different age tissue burdens, depending on whether their diet comes primarily from fresh whole foods prepared at home or primarily from manufactured products processed in industrial conditions. I want to address one more mechanism before the action protocol. And this is the one that connects the dietary age story to the brain specifically, because it is the mechanism most relevant to the cognitive aging that seniors most fear and that most research has failed to explain through the conventional cardiovascular and amyloid pathways alone. The brain has among the highest RAGE receptor density of any organ in the body. Neurons, astrocytes, microglial cells, and the endothelial cells of the blood-brain barrier all express RAGE at high levels. When circulating ages, both dietary and endogenous, reach the brain, RAGE activation in the cerebrovascular endothelium disrupts blood-brain barrier integrity, increasing the permeability that allows inflammatory mediators to enter the neural environment. RAGE activation in microglia triggers the same NF-kappa B inflammatory cascade I described earlier, producing the neuroinflammation that is now recognized as a primary driver of Alzheimer's disease pathology and vascular cognitive impairment. And research has found that the same amyloid beta peptides that form Alzheimer's plaques are age-modified in the brains of Alzheimer's patients, meaning the amyloid has undergone glycation that makes it more resistant to the clearance mechanisms that normally remove amyloid from neural tissue. The dietary age story is not only a cardiovascular and metabolic story. It is a cognitive aging story. Share this video now, before the action plan, with the person in your life who grills every night and has been noticing the cognitive changes they are attributing to normal aging. Share it now while the mechanism connecting their grill to their brain is specific enough to feel urgent. Then come back for the protocol that changes how every meal is prepared. Here is what to do in the next 48 hours based on everything you just learned. Today, audit your typical weekly cooking methods. Write down the 5 foods you eat most frequently and how they are typically prepared. For each one, identify whether the preparation method is high age, grilling, broiling, frying, roasting, toasting, or low age, boiling, steaming, poaching, slow cooking with liquid, or eating raw. This single audit will show you where your highest age exposure is coming from and where the substitutions will have the most impact. This week, make 3 specific cooking method substitutions that collectively reduce your dietary age load by an estimated 50 to 70% without requiring any change in what you eat. First, replace grilled or fried chicken, fish, or eggs with poached, steamed, or slow-cooked preparations at least 4 days per week. A slow cooker or instant pot used on its low-pressure steam setting is the single most practical tool for dramatically reducing dietary age content of animal proteins. Second, replace toasted bread with untoasted bread at breakfast, or replace bread entirely with a starchy vegetable like a boiled or steamed potato, which generates substantially fewer ages than any baked grain product. Third, replace roasted nuts with raw nuts as your primary snack nut. Roasted almonds generate approximately 6 times the ages of raw almonds at equivalent serving sizes. The protein, fat, and fiber content is nearly identical. The age difference is substantial. For your next grocery shop, add acid to your high-heat cooking when you do grill or roast. Research has found that marinating proteins in acidic solutions, lemon juice, vinegar, yogurt, before cooking reduces age formation during subsequent high-heat preparation by approximately 50%. The acid lowers the pH of the protein surface, reducing the reactivity of amino groups toward the carbonyl compounds that drive Maillard reaction age formation. A chicken breast, marinated in lemon juice and olive oil for 2 hours hours before grilling generates approximately half the ages of an unmarinated breast, grilled at the same temperature for the same duration. This is the modification that allows you to retain occasional grilled food without completely abandoning the cooking methods that produce flavors many seniors find most satisfying. At your next physician appointment, ask for a carboxymethyllysine or serum Ag measurement if available at your laboratory, or ask for an alternative marker of age burden, such as skin autofluorescence measurement if your practice has a reader. Skin autofluorescence, the amount of fluorescent age compounds detectable in the skin using a non-invasive device placed on the forearm, is increasingly recognized as a superior predictor of cardiovascular and renal outcomes compared to standard lipid and glucose panels in older adults, and several commercial devices now make it available in clinical and research settings. Ask your physician about it. You now know that the browning, the crust, the caramelization, and the charring that makes cooked food palatable is the same chemistry that is binding to receptors on your cells, activating the inflammatory cascade that accelerates their aging, impairing the mitochondria that power them, and shortening the telomeres that determine how many divisions they have left. You did not know that the smell of grilling meat was a cellular aging signal. You do now. The food has not changed. The cooking method has. And changing the cooking method changes the cellular environment that your body wakes up to every morning. Subscribe to this channel if you have not already. My next video covers the 5 specific foods with the strongest published evidence for directly lowering circulating age burden. The compounds that either block RAGE activation, accelerate age clearance, or inhibit the Maillard reaction in the tissue environment. This is the positive companion to what I described today. Subscribe so it comes directly to you. I am Dr. Edward. Your cells are counting every meal. Make the next one count in the right direction. I will see you in the next one.